Hello and welcome back to Survey of Biology in Unit 12, and we're on Section 4. So we're going to wrap up the immune system today, and what we're going to look at specifically is T cells and how they take care of cells that have already been infected within our body. So as a quick review, if we look at the diagram here, if we start over on the right with the innate immune system, here's the first two lines of defense that we talked about. We talked about the first line, and those are the physical barriers. So that's how we're going to try to keep it out in the first place. So we're going to stop the infection before it enters the body. Then we talked about the second line of defense and how once it's in the body, what can be done. So we talked about the phagocytes, the white blood cells like macrophages and natural killer cells. We talked about the protein complement and the inflammatory response there. And then section 12.3, the one that we just finished, we talked about B-cell immunity, so humoral or antibody-mediated immunity, and how that produces the antibodies and how that takes care of pathogens that are still in the bloodstream. So what we need to look at today is over here on the left, the T-cell immunity or the cell-mediated immunity. And what this does, this goes after the body cells that have already been infected. So this is one of the reasons why you feel kind of crummy when you get sick, because you've got cells that are dying. Your body cells that have been infected are being attacked by your immune system and taken care of. So we're going to focus in on the T cells, and specifically the cytotoxic T cells that will go after and kill infected cells. So if we look at it as a quick review, we talked about how these white blood cells are produced in the bone marrow, and you kind of have a stem cell, undifferentiated, and they're produced in the bone marrow. And some cells will stay there and mature there, and those are the B cells that we talked about last time. So for the antibody-mediated response, with the B cells. Some of these cells will go to the thymus and mature there, and those become the T cells that are part of the cell-mediated response that we're going to talk about today. So if we look at this diagram and go through, this kind of shows the big picture. And what we talked about last time is over here on the left. So here's the humoral or the antibody-mediated. So we talked about how B cells can differentiate into plasma cells and produce antibodies, and then we also get memory cells. So we're going to look over on the right side of the cell-mediated. And what I want to do is I want to actually take a step back and look at how the whole process may start. So let's look at our antigen or pathogen. So the first time that we're exposed to it, it gets engulfed by a macrophage. And what can happen is that macrophage can then take that pathogen, digest it, and actually kind of displays parts of it on the outside. It puts them on the outside of the cell membrane. And I'm going to skip ahead real quick. I guess I'll stay here. And it becomes what's known as an APC, an antigen presenting cell. And basically that has a wanted poster. It has a search image of what the pathogen should look like. And so it will then go to cytotoxic T cells and these things called helper T cells. And those two then get the image from this APC of what they're looking for. This is why the third line of defense is specific, because those cells know specifically what they're going after. So let's see what this really looks like. So here's a picture of exocytosis of an APC. Okay, so exo means to the outside. So what's going to happen is, if we start in this diagram, here's a picture of a macrophage engulfing part of that protein. And if we follow it over to the left, that little bubble, it eats it right up, pinches it off, and will digest it inside. Now, part of that antigen or pathogen that's been digested, that macrophage will then present on the outside of the cell membrane right here. So that's the search image that this antigen presenting cell, this APC, can now take around and show other body cells. Sometimes it'll be the B cells in the antibody mediated response, but in many cases it'll be a helper T cell or a cytotoxic T cell in the cell mediated response. So here's what happens. So here's our APC. So they have engulfed some of those antigens. They're now displaying them on the outside. So they can go around from cell to cell. And in this flow chart, the first one that it shows that to is a helper T cell. And so with that receptor, it locks onto the APC and says, oh, okay, here's what I want to look for. And so what happens is there are these compounds, chemical messages called interleukins. and the antigen presenting cell will release interleukin 1, and that activates the helper T cells. The helper T cells, as I said, can then spread out through the body, and they can activate B cells that we talked about last section, but they can also activate the T cells right over here. That's what we're going to look at here. And the chemical messenger interleukin 2 stimulates the cell division so that we get more of these T cells. And what specifically we're going to talk about after the helper T cell is the cytotoxic T cells. So these are ones that will carry out attacks on infected cells. So these are going to take care of cells in our body that have already been infected. And so that's why it's called cell-mediated immunity, or the T cell immunity, because it's going after viruses and bacteria within infected body cells. So we're going to take care of cells that have already been infected. And this is why people who get organ transplants and people with cancer, the immune system really goes after those, because the body identifies them as being non-self. They don't recognize the proteins that are displayed on the outside of them. So the T cell receptors never secreted. So in the B cells, we saw how those receptors were made into antibodies and dumped into the blood cells. In T cell mediated immunity, the receptors are never secreted. They stay on the cell because it's going to hunt down and track down and hunt the pathogens. So again, here's our cytotoxic T cell. Here's another kind of diagram. And what it will do is it will look for that antigen fragment. It goes and looks for the search image. And when it finds an infected cell that's displaying that fragment, it latches on. It literally grabs hold of it here, and it releases a molecule called perforin. And kind of like the perforations on the side of your paper, the perforin goes and disrupts ion channels and makes water flow into the cell to the point where it will lyse and pop. So this is how cytotoxic T cells kill the infected cells. They latch on, they identify it, they release a molecule called perforin, which causes the molecule to swell, lyse, and burst. And so what we'll end up with is just a quick side-by-side comparison of the two branches of the third line of defense of the immune system. So the first line over on the left, we've got the cell-mediated response. So here's where T cells make contact and they release the granules, and they will then go out and find infected cells. They release the perforin, and they destroy and lyse. So they're taking care of infected cells. And over on the right, we have the humoral or antibody mediated. So here's where B cells will produce the antibodies that will float through the bloodstream. They will bind to the antigen or pathogen, which allows it to kind of slow down so that neutrophils, macrophages, and other white blood cells can come in and gobble them up and remove them from the bloodstream before they can infect any new cells. So there's the immune system in a nutshell. Again, thank you for your time, and we'll talk to you soon.